coming up to the question and answer session for the evening. I'm Ian Wolfe. I do Diffusion Science Radio, which is up there behind me. It's a weekly science and technology podcast at diffusionradio.com. Have a listen. I've interviewed all four of the guests here tonight. So if you want to hear more than you heard in their amazing talks, you can hear them at length in conversation. So the idea here is, if you would take that, that's great. I will just quickly grab. So I've been asked to seed the questions. I think you've all got plenty of questions, but I will seed the questions with a few of my own quickly. So, Vipul, you were saying that the bioplastics can compost the way that they're advertised, but only under very special circumstances. Would you be able to briefly say what those are? To the microphone.
Okay. Yeah, so...
Hold it close.
Yeah, so I can say the name, but I will give you the conditions. So one of the primary ones, which is majority of the industry is using, it requires 40 degrees centigrade at a specific humidity level before it starts to break down. So to achieve 40 degrees centigrade as a uniform temperature on a landfill, I'm sure, I don't know if you have seen a landfill, they're kind of like mines. They're similar kind of size, they're huge. So if you can imagine maintaining 40 degrees in that open pit, it's kind of very challenging thing and a very expensive thing as well. So so it's not practical, and that's why it doesn't serve its true purpose in that sense. If I can say that, it's a contentious comment, but that's what it means like.
That's right. I led you into that one. Okay, so I've got a question for Helen. You mentioned briefly the new curriculum will be coming in. Would you be able to say briefly how it's different?
I think I'm not allowed to say how it's different, actually.
Secret.
However, the last draft revealed that it's not actually that different. So the aim was to make minor changes and get feedback to really optimise different aspects of it. But as with all curriculum reform, there are still contentious issues with this curriculum. So you'll just have to tune in for the next segment of this story.
Fair enough. And I'll try that. No, I think, we've got it. Question for you, Liz. You mentioned in the reproduction, here we go, here's for you to be able to answer. In the reproduction of bacterial cells, you talked about in the middle of the cell there was a spindle. Why is it called a spindle?
So in bacterial cells they have a polymer. That the spindle is actually the mitotic spindle of tubulin in our cells. Yeah. So it's a good question. My guess is that it's because it grows from a point and polymers, if you like, protein polymers, different type of polymer, are actually added to it, and then they start to contract away by joining the filaments. So I guess that's why it's called a spindle.
Thank you. And question for Benjamin. Could you tell us a little bit more about Ruby Payne-Scott?
Very, very happily. Okay, so Ruby Payne-Scott, my idol, was an Australian astronomer who sort of flourished in the 1940s and early 50s. She had a remarkable career. So she studied at the University of Sydney. She, Sydney girl, she went to Cleveland Street Girls, then Sydney Girls, then University of Sydney. And she did her master's at the University of Sydney because there were no opportunities for a woman to continue on to a PhD at that point in time. Right? And so she ended up working for AWA, the Australian Amalgamated Wireless Australasia. What's the actual acronym? So it was the radio manufacturer. It's the sort of Eiffel Tower next to Wynyard, right? It used to be Australia's largest electronics manufacturing firm. And she ended up originally hired as a librarian, and then they quickly realised that she had great technical skill and swapped her onto the engineering. And, one of the things... it's very popular to talk about hidden figures stories, which I think are super, super important. Ruby Payne-Scott is not merely Australia's one of those. She is very strongly, her whole career has been shaped by sexism, right? And so, she had to fight for the right to wear shorts at work when she was installing antennas in, hot conditions in Sydney. Anyway, she flourished in her career there and then was at the CSIR, before it was the CSIRO, at Dover Heights. So it's now, if you've done the Coast Walk, it's actually a football field, belongs to the school there, which is, I think, disgraceful because this is actually one of Australia's most important scientific sites. Because during the war, it was a radar station. And they observed, the idea was to look for Japanese aircraft coming, or ships coming towards Sydney Heads. But after the war, following up work by a New Zealand woman, Elizabeth Alexander, she actually observed the sun with radar. And she took the first radio image. That's the thing that she is rightfully really famous for, is using the first measurement of radio interference to make an image. So this is the science of interferometry. And so actually the interferometry, normally you need multiple telescopes and you combine them, but she only had one. What she actually did is she used the telescope and its image reflected in the sea as two components. So it's the Seacliff interferometer. They had one at, they did this experiment first at Dover Heights and then also at Collaroy. So you can actually go up to Collaroy and you can see it on the cliffs there. There's a little plaque, I think, commemorates it. And they measured, in fact, that the sun was not merely emitting radio waves, but these were coming from sunspots. And her team discovered all 4 types of solar radio burst, which are the kinds of radio emission you get from solar flares. So there's 4 types. She named 2. People under her supervision named the other 2. And so really you're talking about someone who is probably Australia's most impactful scientist of that decade, right? It's very hard to think of someone else in the 40s, certainly in physics, with anywhere near, within an order of magnitude, the same impact. And so she gets married. And the good news is out of that relationship, this guy, Bill Hall, gets Peter Hall, is her son, and he ends up being the eponym of the statistics department at University of Melbourne, a very prominent Australian statistician. And Fiona Hall, the artist, is the daughter. And so she's got the big sculpture in the Botanic Gardens of that sort of cage thing next to Government House. That's Fiona Hall, among many other things. She's probably, at the peak of her career, she was probably Australia's leading sculptor. There's the good news. The bad news is because she had kids, she was caught out for being married. It was rather hard to hide that fact. And it was at the time illegal to be a married woman in the Australian Commonwealth Public Service. And so there was an enormous fight over what to do about this. And Ian Clunies-Ross, the head of the CSIRO, personally made sure that she was fired. And she spent the rest of her career teaching maths, which is a great and noble profession, and I'm sure many people in this room do that, but she didn't want to. And by all accounts, wasn't really keen on the whole thing. And that was that. And so this was this really remarkable story of a leading, Australian scientist whose career was destroyed for this, but whose invention of interferometry and whose study of the solar activity in radio is completely foundational, not just to modern radio astronomy, but to things like MRI and Wi-Fi. Really, Ruby Payne-Scott's innovation in developing radio interferometry was the thing that had made Australia the leading centre of radio astronomy that it is today, that it was throughout the mid-20th century, and why Australian scientists are still leading this discipline today is because of the intellectual heritage coming from that.
Thank you, Benjamin. Okay, so we're opening it up to the audience. So if you wish to ask questions, you're putting your hand up so that we can have someone bring a microphone to you. Oh, if we can, yes, the microphone coming around. Oh, we've got someone over here as well. We've got someone over here as for the next one. You'll be next then, okay? So one over here, if you wish to say who you're addressing your question to, and just one question at a time, please.
Okay. First, I was going to ask Helen, but just on Helen Payne, Ruby Payne-Scott. You also know that she was under investigation, as many scientists were at Sydney University, by ASIO.
She was a communist, overtly a communist.
My question is to Helen, because my name is Peter Farley and I'm the education officer at ANSTO. So So I take lots and lots of high school kids through and teach them radiation, physics, etc. Our Discovery Centre had 17,000 visitors last year. That's 16,000 year nines. Well, not quite, it felt like that. Nevertheless, with the two curriculum, to have something that has got a social basis and then one comparative with one with more rigor, you've got to have kind of both, and that's what they try to do now in the senior years with the investigating science program. The trouble is with the it's seen as something to kind of get points, and the emphasis that I try to give the kids that do that, because I actually specialise, because not only was I have an electrical engineering background, but I have a philosophy degree as well, and taught HPS at Sydney Uni, is that you've got to say that these kids that the importance is to straddle the two cultures. So they may go on to be lawyers or politicians or some sort of policy makers, but then they've got to have some basic understanding of science and not to be afraid of science and to know who to go to speak to about it. So is that not the way to go, to try and have both in the curriculum?
Yes, that would appear to be the best solution to this problem. However, that's just not how a lot of education works. So, and I'm going to sort of go into sociology again. What tends to happen with people is that they take either one side or the other side. And this has resulted in a number of like big dichotomies in education. So you would have heard of some of them. They're at the centre of like the reading wars currently in science. It's sort of like explicit instruction against inquiry-based approaches, and it's the same with the syllabus. It simply can't be both. It has to be one or the other, just because of the way people sort of work and how those narratives come out. Interestingly, with HSC subjects, so as you say, when we try to go to something like investigating science, what happens is that it has a lower value assigned to it. So sociologically, you can say it has been charged with like a negative value. And we see that in the way that the ATAR is scaled, and we also see a relationship between ATAR scaling and socioeconomic background of students. So once it comes out and there's a signal, this is a lower value subject, then it's not a subject that those higher students are taking, and it becomes relegated to like this second place. And so we haven't been able to find that balance of like, can it still be a high value subject, but include those elements that we think are important to include like for science curricula.
Thank you. Okay, question over here. Who is it for, and what's the question, please?
Question for Vipul. How far away is the commercialisation of your technology?
That's a wonderful question. I would like to say tomorrow, but I can't because of the limited funding. However, we are quite advanced in our PET recycling and commercialization journey. Soft plastics, we are still awaiting some sort of substantial funding for us to keep continuing. But we believe that I would like to give myself a 5-year challenge to transfer the red bin into a yellow bin. That's the holy grail. That's what I would like to achieve, because right now we cannot. So long story short, soft plastics, not sure, because there's no appetite from government or from industry right now to support. We are actively seeking support, so let's hope there'll be something soon. But for the hard plastic, which is PET, hopefully within the next few years, you will start using them in your confectionery at the very least.
Fingers crossed.
All right. So we have a question over here. Oh, down here.
So my question is for Helen. PISA's been around for a long time, and I have to admit I took quite a bit of interest in it back in the 80s. So my memory may be failing me. But what bothered me when I looked at it then was it's all about the test, and the claims are made that you can design a test that is corrected for cultural differences, and which is then able to intercompare everybody from these disparate cultural backgrounds. And I'm not sure that I was ever read anything that convinced me that that can actually be done. And yet the results continue to be reported without any mention of what they're actually based on test-wise and how that intercomparison is done. So should we really be giving it the weight that we give it is part of the question. Secondly, you've talked about the TIMS test. If that is a test where we get to test ourselves against our own curriculum, then it's not really any sort of international comparison at all. We're setting our test standard and we get the result that we get. So the testing seems to me to be problematic. And do they in PISA now actually interrogate whether or not kids like it? Because back when I did look at it, Hungary and Japan were top of the heap at that time. But there was also research at the time that interrogated the attitude of the kids to the subject. And the Japanese kids didn't like physics, even though they did really, really well at it. And so shouldn't we be concerned about that?
Yeah, definitely. So I'll try to remember back to the first question. PISA does have an issue, as with all of these tests, with validity. Like, are they testing the thing that they say they are testing? And for PISA, I think the answer is no. So when I looked at the research into, you know, why were these scores declining and not TIMS? What people found was that there was a correlation between all of those scores. So they were all declining, but research showed that they were all related. So you couldn't say that science literacy was different to mathematics literacy and even reading literacy. So they were testing essentially the same underlying construct, not distinct constructs. So there's an issue there with validity of the tests, for sure. What was the other question that you...
I mean, nationally we're setting our own standard, and so we can bias it. So
they have ways. They have ways of standardising it across. So they use the same items to then compare samples to then match them. So they've got ways of sort of doing that, and curricula are not that different. So actually you can have kind of a broad set of the same questions that lots of students do see. But the analysis of these tests is like, people think it's like a very exact science, but it's not really. There's a lot of things that people are doing that are just a little bit dodgy or a little bit open to lots of different, interpretations. So you've just got to be careful. Like, we've got to be careful. And no, I remember your question now. We shouldn't be paying the attention that we do to PISA. I think like over 90% of references to standardised tests in Australia are about PISA, and we're like number 2 in terms of the mentions of PISA. So we are obsessed with PISA. We've got to let it go a little bit. No other country is this, obsessed, except for the United States. And so, yeah, we've got to just kind of be a little bit more varied in our consideration of the different forms of evidence.
So perhaps that could be a press release by the AIP.
Yeah. Well, I tried. People are not like that interested in like saying we're doing good, we haven't done any different, and things are not that bad. It's not a sexy headline.
Thank you. Okay, so we've got the next question over here. If you say who it's for and not too many questions.
This is for Professor Harry. How does the time that it takes to replicate the organism, how is the time to replicate the organism important when it comes to replicating it? And are there specific things that must be followed for it to be fully replicated?
Is this working?
Yeah. I'll try this one.
So this is exactly the research question I've been studying all my career. So bacteria replicate. It depends where they are and what the bacterium is. But I always joke about studying bacteria because I'm impatient, and I study bacteria because they divide very quickly. You don't have to wait long to take a single colony, put it in a broth, usually do an overnight culture, but the next day, in a couple of hours, you've got enough cells to do your experiments. So in us and in infection, they can, once they get going, they can multiply very quickly. So in the lab, for example, they can double their mass in 10 minutes, right, at 37 degrees. Not all bacteria, but the one I studied, and E. coli, can do it very quickly. And it's interesting how they do this, because the chromosome replication process from one, the whole chromosome to be replicated takes about 40 minutes. So how does this happen? And this is why it's so hard to study how the cell manages to grow and end up, when it stops growing, with each cell having one piece of DNA in it. So what happens is it starts a round of replication, and then it'll start another round before that first round's finished. Yeah? And then once it starts a round, it says to the division machinery, okay, place yourselves there and wait until we've done enough replication and chromosome segregation to start dividing, right? So there's signals all the way along. And then there's another initiation event or another starting of DNA replication that happens at another place, and then it sets up division again. So you've got these cycles going on all the time. With human cells, most of the time it's DNA replication, DNA replication, separation of chromosomes, separate into division, separate cells, and then you have a growth period. And they have to go through those stages. So it's a really good question, because it's been very hard to work out how do bacteria do this.
You also mentioned that if it's replicated too fast, both the host cell and the replicated cell dies. Why is that?
So I think you're referring to when we have bacteria inside the host cell. Is that what you're referring to? How does it die?
No,
sorry.
When the bacteria replicates, you said that if it's done too fast, both the host bacterium and the bacterium that was supposed to be replicated dies. And how does that speed refer to how it can survive?
I don't think the bacteria necessarily dies when it bursts. When it kills the host cell, it usually doesn't die. Yeah, it might, but it usually doesn't die. Okay, and then it goes in and gets into other cells, just like a virus, as we were talking before. Yeah. Thank you for your question.
Thank you. Okay, we've got one over here.
This is a question from Professor Harry Exton. My question has two parts, actually. Firstly, it's about the discovery of the FTZ, and as I think it actually conjoins with the previous question about the specific protein actually moves with the time and space. Now, the first part is about, can we have a flame-based, fluorescence lifetime imaging theranostic agent that actually dually target the FTZ at the same time the potassium channel blocker in such a way so that you could monitor it at one particular point of time, like where the FTZ actually moves on, and how does that impact with the potassium channel blocking? That's one thing. And then this it's actually the second part is about can we, if we have that sort of ideally, if we have that sort of that dual targeted or multi-targeted flame-based theranostic, then can we correlate with that quorum sensing of a natural product like curcumin? So I'm just.
So when you mention potassium channels, why are you referring to that? I'm not sure what you're.
Yes, that is because in UTI, Klippel pneumonia is actually, and it actually mostly related to the potassium channel blocking. So if you this could be a nice. And on the third point, that would be like if we have some answers for these, and can you correlate the way the bacterial cell that divides to the tumor cell division, so that actually will give us some sort of replication and correlation. Can there be, because we have now proven that there's a microbial infection and cancer has a very strong correlation from the perspective of Alzheimer's, and it goes back to it.
So the short answer is you should be able to do all of that. Yeah. So yeah, you should be able to. Basically, you can follow FTSZ. You can follow the division proteins. You can label them all in live cells. You can follow them. You can check that label doesn't affect, obviously, their survival, but also the function of that protein. That's sometimes a challenge, because FDS said it's hard to get a functional version of some of these proteins if they polymerise. Or, it's amazing that you can get functional proteins when you add a big other protein to them, but you can generally, and if you can't label FTSZ, you can usually label something else that we know goes to the same place. So yeah, you can basically do all of that. Yep. All possible. Yep. And they're good ideas. Yeah.
Fantastic. So we've got John. Who do you want to ask the question, and what is your
Liz
question?
Is my question for you. Had a graph with the sizes of different bacteria, and you had one there, E. coli, and you had one that from your graph it seemed to be about a tenth of that size. So I guess my question is about really small bacteria. Are they just smaller versions of regular sized bacteria, or do they have something different about them at some more fundamental level?
Yeah, that's a really good question. So Mycoplasma genitalium and other mycoplasmas also cause problems for animals and humans, so not all of them, but they're bad infectious agents. And one of the guys in our institute is studying those as well. So yes, they tend to have a reduced number of genes and therefore number of proteins that do things. So those cells also have don't really have a cell wall, which is again how we used to classify bacteria, is they have this cell wall made of a very thick substance that gives the cells their shape. So these mycoplasmids are little round cells, and yes, they're about 0.2 microns, so they're tiny. Yeah, so they're quite different. They still have FTSZ, and they still divide in a similar way, but yeah, they're minimal genomes. They just probably happen by chance, like evolution does, and they happen to find a niche. Yeah, thank you.
And we've got another question here. Who do you want to ask the question of? And
yeah, just wondering, with the image reconstruction or the image cleaning technique that you have, is it based on more a classical approach, like maximum method, Bayesian approach? Or are you able to use something like an artificial intelligence method, like a deep learning, something like that to assist?
All of the above is the short answer. So the question sort of refers to if you've got these data, which constrain some image, they don't actually give you this image unambiguously. They constrain, if you're familiar with the Fourier transform, or even if you're familiar with JPEGs, how you compress images, they constrain a few coefficients of that representation of an image, but they don't constrain the whole thing, right? And so there are infinitely many possible images. You can paint as many angels dancing on the head of a pin as you want in the gaps between all of these coefficients, right? And so you have to have some way of in-painting those. And so there are classical methods that were sort of developed in the 80s, which is why I think it's funny when you get in arguments with people. They say, you absolutely can't deconvolve images. I say, you absolutely can. You have since before I was born, right? These methods, you say there's some kind of naturalness metric about an image. You say that images might be smooth, or they might consist of edges separating regions that are relatively uniform. Or you might say, I want to find an image that has the maximum entropy, which means the least additional information has to be specified beyond what we've already got to specify this image. And therefore, it's the most agnostic possible image. So you have all these regularized image reconstruction, you call it methods, where you can say, I got these data, the data don't really tell me unambiguously the image, but I know something else that's natural about the image. So we're doing that. And so that's what my student's going to do for his paper coming up. On the other hand, they're not nearly as good as methods using artificial intelligence. And so we are also working on another paper where we're using what's called a score-based prior, which is the technical term for what's sort of behind the stable diffusion. Or if you log onto your favorite website and you have a picture of a cat wearing a hat, is what you ask it for. It'll use stable diffusion. And a score-based prior is the idea that real images live in some probability distribution that is very, very hard to describe. But a score is an archaic, and frankly, I don't like it, term for the gradient of the logarithm of a probability distribution. And it turns out that it's much easier to learn that than it is to learn a probability distribution itself. And so there are neural networks that can well approximate not how likely is this image, but how much more likely is this image than a very similar nearby image. So that's the sense in which this is a score-based thing, is it can tell you locally how much more likely things are one to another. And so we're also writing a paper using score-based priors to deconvolve these, and that's sort of the current state-of-the-art in AI image generation.
Okay. We have a question here.
Question for all the panel members. So I'm a science teacher, and I was just wondering, you've all gone into science and different fields, but I'm assuming a similar love for what you do stemmed from maybe a teacher or an experience at school. So just wondering if you could briefly mention what started the journey, and maybe we can leave with that.
I'm happy to. I've got the microphone, so I'll just pass it down. Yes, it unambiguously came from a single science teacher. Some of you may have known him. He was relatively prominent. Michael Bishop was his name. He was a chemist. One of the most inspiring people I've ever met. And I was mainly interested in languages and literature at school until I started learning from Michael Bishop. And I think actually, I mean, the thing that really, what really impressed me the value of being a scientist from him is the way that you could be many things all at once. He taught us chemistry, he taught us physics, and he also taught us Viking and Egyptian literature, right? And so, you know, I think we can focus too much on STEM education. I think we can focus too much on particular deliverable outcomes in narrow sense STEM, whereas in fact I think it's a way of looking at the world, and you can look at a lot of things in the world that aren't just physics and aren't just chemistry in this way, and that's what he taught me. And it made me realise that I could be the kind of person that he was, or at least I would like to try, and that being open to the world in a more general sense was an option.
It's a good question because I'm not really sure. I think part of it, I was born with a curiosity, and I think I was born in Manly, and I love the beach. I still do. Water is means so much to me, and I used to spend a lot of time just walking around the rock pools and looking at the periwinkles and the conjovoids and the squirty seaweedy stuff that used to. You're not meant to do this, but we used to press on it. Water used to come out. That was a lot of fun, and we used to squirt it at each other. I also couldn't do this now, but I used to ride my bike at Dobroyd Point because friends used to live around that area, so all that national park. So I was surrounded by that, a lot of bush. So maybe that's what it is. I did have a good biology teacher, and she was very amusing, and I recently went up with her again. But yeah, we got on really well. But yeah, I think it was mainly just the surrounds.
I feel really awkward saying this because I'm in the job of like inspiring others to love STEM, but I feel like it was always there for me. So I was first in family university. No one else in my family had gone to university. In fact, lots of my family didn't finish school. They migrated over from Europe. I had no mentor or kind of vision of what someone looked like to go through and select a career even. But I remember like books in the home and encouragement, and back when WH&S wasn't an issue, I had a children's book which, showed me how to use a magnifying glass to set a leaf on fire. And I just remember being like just absolutely enthralled by these things. And it was that kind of inherent thing that was then, yes, definitely buoyed by wonderful teachers, wonderful supervisors who have got here today, and family members and friends, and like Ben and I were doing our PhDs at the same time, so people in science that I just was really drawn to, and all of those things kind of coming together. And I wish I had a better idea of it, because then I could help others more, but it is a very complicated thing and different for everyone. Good question.
Right, thank you. Yeah, very interesting question. Somebody asked me that question before as well. I still do not know the answer to that question. I think for us, being an Indian, it's a cultural thing. So I remember, especially after when I decided to do my undergrad, I wanted to do literature. And my parents were like, well, what are you going to do after literature? You're not going to have a job. So the only profession that can get you a job, at least back in the day, my parents' thinking was become an engineer or a doctor. And I didn't become either of those two. But I still teach engineers. So it's very interesting. But I still remember, I think because the societal push is so much towards science, and there was a narrow thinking, at least back in the day, that is where you can get a job and have a living. My dad was a doctor, so he wanted us, at least, I've got 3 siblings, so one of us to become a doctor. None of us became a doctor. So that was pretty disappointing for him.
Still a doctor.
What doctorate? He's a medical doctor, so it's a higher bar apparently. So I think what happened was I was naturally curious reading what is called, I don't know if you have it here, it's called Reader's Digest. It used to be a small, really, really interesting thing. And we didn't have a television at home, but my mom's friend used to have a television. So I would go there, and they used to have a cable, and I really loved National Geographic and Discovery. It was just so fascinating looking at oceans and animals, and it just drove me into something. So I don't know if that is what precipitated into it, because when I went to university, I was terrible. I did honors in chemistry. I was terrible in the first year. I had no idea what I was doing. It took a long time before I got accustomed to it. Same thing when I moved the country and started doing my master's. And that could be a deterrent at the start, right? Because you start self-doubting, am I good enough to do this? And honestly, you are, if you persevere. Science is all about perseverance. That's it. That's what it's all about. So I think kids can be inspired that way, but the environment around them makes a huge difference. I can give you, if I have time, like one more example. So I was invited to talk to, I think year 6 to year 9 students in one of the remote schools in New South Wales. And I was sitting on a table about career night, and I was talking to them, and I asked them, what do you want to do after, high school? And one of the kids was like, I want to be a plumber. So there's nothing wrong with being a plumber. And I was like, but do you know there are other things you can do? I was like, well, you can start earning money straight away. Right? Like that's the motivation. I was like, if you go to university, you can still earn the money, and you will get, a lot of money over time. So he was like, well, how much do you make? And I was like, it's not about the amount of money. It's about the skill set, and you can go anywhere in the world and still, make money. So it's about, I think it comes from teachers, but it also comes from the home environment.
Thank you. We've got the last one over here. Oh, here we go.
Yeah, just
sorry.
A question for Vipul. So, what is the current cost to having a landfill, maintaining it? I guess like finding new land for them, as opposed to recycling the plastics. Is it having landfills actually more expensive, and recycling them is actually cheaper? But because we don't really have an effective way to recycle plastics, so we stick with landfills. And to follow up to that is, how is your technology going to substantially reduce the cost of making this economically viable?
I think with the landfill, it's a necessity. Because it's the limitation of the technologies that we currently have. We can only recycle plastic few times before it has to go to the landfill, right? So the cost is a factor, but the factor is not because landfills is cheaper versus recycling. It's because recycling cannot deal with the requirements of the industry. That is the problem, and it has to go to landfill, right? So in our technology, we believe that we can recycle it way more, and I can't put a number, and I don't like to use the word infinity, even though I've been asked by other people to use that word. There is no such thing as infinity in terms of chemistry, so you cannot recycle something infinite times, but I believe that we can recycle it way more number of times than we can currently, and therefore the demand for landfill would not only be delayed, but it would be reduced quite substantially. So for me, biggest challenge is actually closing the loop. So people use this word very loosely if you start talking to them about it. Closing the loop means you reduce the need for virgin plastic. That is what recycling should be like. So you reduce the need for virgin plastic and just keep using what you already have in the system, which is thousands of tons, if not tens of thousands of tons or hundreds of thousands of tons of plastic already in circulation. Right? So if we can do that, then we don't need landfills. Right? So I hope I answered your question. Thank you.
Well, perhaps afterwards. Well, thank you, everybody. Oh, no, what have we got left? We've got our next, that's the end of the question and answers? Or we got one more question? I'm trying to understand. That's it. Thank you, everybody, for all those really interesting questions. And now we're on to the last part of the evening.